part we'll be discussing about the pollination in case of Cycas. Pollination here is anemophilous in the sense the pollen grains are to be carried out by the wind, air currents. Such pollination is called as anemophilous. The cells of the nuclear beak, this is the pollen chamber, this white small area, and this will be the archegonial chamber from which the archegonia is going to develop. So the cells present in this nuclear beak are going to disorganize and lead to formation of a fluid, viscous fluid. This viscous fluid is going to ooze out and it is going to settle down at this point. This fluid which is going to ooze out and settle down here, that fluid is called as a pollination drop. Yeah, this picture. So we have a ovule here followed by this is that micropyle end. This is the fluid which has oozed out and settled down here. This is sticky in nature. As a result, when the wind is going to carry the pollen grains will be stuck inside this sticky fluid. This is called as a pollination drop. And this pollination drop is going to entangle the pollen grains present in the air current at their 3-celled stage. Gradually the pollen drop dries up and the pollen grains are sucked into the pollen chamber. So this is going to dry up and they are sucked inside this pollen chamber. So when they are going to enter pollen grains, they are going to settle down here in the pollen chamber. And as a result of drying up of the pollen drop at this tip, it is going to seal this open micropyle end. Seal in the sense it is going to form a lid-like structure. So no more entry to the pollen grains. Now the pollen grains are entered in the pollen chamber. No more entry for the other pollen grains. It is going to act as a seal at this opening end. So this is the role of a pollen drop, that it is going to entangle the pollen grains and also the seal micropyle end so that there is no further entry for the pollen grains. Now next step will be, we have discussed that these pollen grains show 2 stages of development. One will be in the microsporangia, another will be in the ovule, particularly in the pollen chamber especially. Here, after entering into a pollen chamber, they are going to rest for a while. The resting period may last for some 20 days to 4 months. It is usually 20 days in case of a Cycas revoluta. Further development of this male gametophyte takes place in the pollen chamber. It has got 2 coverings, that is exine as well as the inner intine. Now the exine is going to rupture, that is it is going to break up, and what it leads to, remaining part, that is intine. That intine is going to come out in this manner, in the tube manner. That tube manner is called as pollen tube. So exine is ruptured, it is going to break off. Now intine is going to form a tube. That tube is called as pollen tube. So we have this 3-celled stage: prothelial cell, generative cell and tube cell. This is a stage where it is going to enter into the pollen chamber. So inside pollen chamber, what happens? Exine is going to break off. What we are left with? Intine. Now intine is going to form a tube. This tube is called as a pollen tube. Now next step will be this generative cell. This generative cell is going to divide. This pollen tube is there, no? This pollen tube is going to act as an absorbing organ. We have discussed a fungus also in our first SEM classes. There a word comes, that word is nothing but a hostorium, hostoria or hostorium, which is nothing but an absorbing organ present in the fungus. The similar function is done by this pollen tube. This pollen tube is going to absorb the nutrient. For why? For the development of the sperms. Yet the sperms are not formed. The microspore is in this 3-celled stage. It leads to formation, further development that leads to formation of the sperm. For that it requires the nutrient, and that nutrient is from the ovule only. Ovule is going to provide the nutrient, but the absorbing part is very important. That absorption is done by this pollen tube. So the sperms formed only after the complete development of the pollen tube. Now pollen tube penetrates the nuclear tissue and it lies in this archegonial chamber. It was in a pollen chamber, right? Sperms entered and landed over in the pollen chamber. Exine break off. We are left with the intine, and the intine led to formation of this pollen tube. This pollen tube is going to act as an absorbing organ, and from pollen chamber now it is going to enter this archegonial chamber. When it is going to lie in this archegonial chamber, the generative cell, this generative cell is going to divide, and it leads to formation of the two cell, the upper body cell and the lower stalk cell. This is the upper body cell and this is the lower stalk cell. This stalk cell won't show any further development. That is, the stalk cell does not divide further, whereas the body cell divide, and it lead to formation of what is called as blepharoplast, and followed by another vertical division, and this leads to development of the sperms. Now what exactly happens is this this is the body cell. Now this body cell forms a structure called as blepharoplast, where on its transverse side, and there is a formation of a wall, vertical division, leading to formation of the two cells. Now these two cells are having their respective blepharoplast and a nucleus. So the longitudinal division in the body cell, this body cell is the generative cell we had. This generative cell led to division, leading to formation of the two cells, the upper body cell and the lower stalk cell. The stalk cell won't show further division, whereas the body cell, it led to formation of the blepharoplast on each pole, on each side, on the horizontal side. Further step will be vertical division in this cell, leading to formation of the two cells with each blepharoplast on their cells. Now each of these having a single nucleus and a blepharoplast, and small amount of cytoplasm will also be there. Each of these is going to develop and form what is called as a sperm. Why? Because this blepharoplast is going to give rise to cilia, and this lead to formation of the sperm. These sperms are somewhat top shaped. Top antandre bagri, top shaped structure with somewhat triangular or top shaped structure having spiral bands on them. Likewise we have spiral bands with large amount of thousands of cilia present with a single large nucleus. So it is nothing but a kind of metamorphosis. A tube cell, generative cell, it led to formation of the body cell as well as the stalk cell. Stalk cell won't show further development, whereas the body cell, it metamorphosis, you can say, leading to formation of a blepharoplast on each side, and it led to vertical division. Now vertical division leads to formation of the two cells with a large nucleus and with their own respective blepharoplast. These blepharoplasts are responsible for the formation of the cilia, and they are going to form what is called as sperms. These sperms are usually triangular in shape or top shaped structure with spirally arranged bands on them and having thousands of cilia. So they are motile. So these are motile in nature. Now the pollen tube is going to rupture and leading to formation of a release of these sperms, mature one, into the archegonium. So they will enter the archegonia, and fusion of the male and the female nucleus takes place, leading to formation of a diploid zygote. Here more than two archegonia, chances of like more than two archegonia even may be present in case of Cycas, but usually archegonia will be two in number. So this was some detailed description regarding pollination in case of Cycas. Thank you.